1. Okay, chapter 12, chromosomal basis of inheritance. Mendel's hereditary factors were genes. Today we know that genes are located on chromosomes. The location of a particular gene can be seen by tagging isolated chromosomes with a fluorescent dye that highlights the gene, like so. Mitosis and meiosis were first described in the late 1800s. The chromosome theory of inheritance states that Mendelian genes have specific loci or positions on chromosomes, and chromosomes undergo segregation or separation in independent assortment, where they line up randomly at the metaphase plate and separate into different cells or gametes. The behavior of chromosomes during meiosis can account for Mendel's laws of segregation and independent assortment. So here we're looking at the chromosomes that are undergoing meiosis. Okay, so here we have our P generation. So this is the parental generation. So here we have yellow round seeds, and here we have green wrinkled seeds. And then after meiosis, meiosis halves the chromosome numbers. So here we see haploid cells with one of each type of chromosome in them. Okay, so here are our F1 plants. So they are all yellow and round. So this just follows them through meiosis. So metaphase 1, they line up at the metaphase plate. So look at, there are two possible combinations that they can line up in here. And then anaphase 1, remember that the pairs separate. Metaphase 2, they line up individually at the metaphase plate. And then anaphase 2, they are pulled apart at their centromeres. So here you can see the possible gametes that you can have. Okay, so when we cross two F1s, remember that our phenotypic ratio in our offspring should be 9 to 3 to 3 to 1. This results from the law of segregation and the law of independent assortment. Thomas Hunt Morgan and his students began studying the genetics of the fruit fly, Drosophila melanogaster, in 1907. Several characteristics made fruit flies a convenient organism for these studies. They produce many offspring. Their generation times are really short. A generation can be bred every 2 weeks, and they have only 4 pairs of chromosomes, so they're really easy to study. Morgan noted wild type, or normal, phenotypes that were common in the fly populations. Traits alternative to the wild type are called mutant phenotypes. The first mutant phenotype they discovered was a fly with white eyes instead of the wild type red. Okay, so here we have the red eye and the white eye flies. In one experiment, Morgan mated male flies with white eyes, the mutants, and female flies with red eyes, which was the wild type. The F1 generation all had red eyes. The F2 generation showed the classical 3-to-1 red to white ratio, but only males had white eyes. So Morgan concluded that the white eyes had to do with the sex of the fly. Morgan determined that the white-eyed mutant allele must be located on the X chromosome. Morgan's findings supported the chromosomal theory of inheritance. Okay, so in a cross between a wild-type female fruit fly and a mutant white-eyed male, what color eyes will the F1 and the F2 offspring have? So here, the F1 offspring, these are the ones that they all had red eyes. And then for the F2 generation, this is just showing that the females had red eyes and one of the males had a red eye, versus the white eyes were only found in males. So 3-to-1 ratio. But sex was a factor in this. In humans and some other animals, there's a chromosomal basis of sex determination. In humans and other mammals, there are 2 varieties of sex chromosomes, a larger X chromosome and a smaller Y chromosome. Only the ends of the Y chromosome have regions that are homologous with corresponding regions of the X chromosome. The SRY gene on the Y chromosome is required for the development of testes. So here's the much larger X chromosome versus the Y chromosome. Females are XX, males are XY. Each female egg or ovum contains an X chromosome, while a sperm may either contain an X or a Y chromosome. So it is the male that will determine the sex of the offspring, because he is the only one that can give a Y chromosome to his offspring, or the zygote. Other animals have different methods of sex determination. So this is male versus female. So we have 44 autosomes and then 2 sex chromosomes. For males, that's XY. For females, that's XX. So when they give chromosomes to their gametes, rather, the males give 22 autosomes plus an X or a Y chromosome, and females automatically give 22 autosomes plus an X chromosome. A gene that is located on either sex chromosome is called a sex-linked gene. Genes on the Y chromosome are called Y-linked genes. There are a few of these. Genes on the X chromosome are called X-linked genes. X chromosomes have genes for many characters unrelated to sex, whereas the Y chromosome mainly encodes genes related to sex determination. X-linked genes follow specific patterns of inheritance. For a recessive X-linked trait to be expressed, a female needs 2 copies of the allele because she has 2 X chromosomes. However, males, they only need 1 copy of the recessive allele because they are XY. So if the recessive trait is on the X chromosome, then they will show the trait or the disorder. As a result of males only needing 1 copy of the recessive allele, X-linked recessive disorders are much more common in males than in females. Some disorders caused by recessive alleles on the X chromosome in humans include color blindness, mostly X-linked, Duchenne muscular dystrophy, and hemophilia. In mammalian females, one of the 2 X chromosomes in each cell is randomly inactivated during embryonic development. The inactive X condenses into a bar body. If a female is heterozygous for a particular gene located on the X chromosome, she will be a mosaic for that character, such as in the tortoiseshell cat. So the tortoiseshell cat has an allele for orange fur and an allele for black fur in every one of her cells. However, in each one of her cells, one of those chromosomes has become inactivated. So in the cells where the orange allele has become inactivated, the cat will have black fur. And in cells where the black allele has been activated, she will have orange fur. Each chromosome has hundreds or thousands of genes except the Y chromosome. Genes located on the same chromosome that tend to be inherited together are called linked genes. Morgan did experiments with fruit flies that show how linkage affects inheritance of 2 characters. Morgan crossed flies that differed in traits of body color and wing size. He found that body color and wing size are usually inherited together in specific combinations. He reasoned that since these genes did not assort independently, they were on the same chromosome. So not only were they on the same chromosome, they had to be close together on the same chromosome, because remember, if they are far apart, then those genes are the chromosomes are gonna cross over. So those alleles most likely will be separated at least 50% of the time. Okay. Oh, this is really small. Okay, so here we have our parental generation. So the parental generation was the wild type was gray body, normal wings, and the double mutant was black body and vestigial wings. And then they were crossed together. And what we should see, if genes sort independently, is a 1-to-1 to 1-to-1 ratio. The genes would sort independently if they're on different chromosomes. Now, if the genes are on the same chromosome, our expected ratio would be 1 wild type to 1 black vestigial, which is the double mutant. These would match the parents, okay? Now, when they actually did this experiment, they got 965 of the wild type, 944 of the black vestigial, but then they also got 206 gray vestigial, which is one of the recombinant types, and 185 black normal, which is the other recombinant type. So what happened here was that genes were close together on the same chromosome, but they did show a small amount of crossing over. So, what they saw was mostly parental phenotypes, but they also saw that there were some non-parental phenotypes. Understanding this result involves exploring genetic recombination, the production of offspring with combination of traits differing from either parent. The genetic findings of Mendel and Morgan relate to the chromosomal basis of recombination. Mendel observed that combinations of traits in some offspring differ from either parent. Offspring with a phenotype matching one of the parental phenotypes are called parental types. Offspring with non-parental phenotypes are called recombinant types or recombinants. A 50% frequency of recombination is observed for any two genes on different chromosomes. Morgan discovered that even when two genes were on the same chromosome, some recombinant phenotypes were observed. He proposed that some process must occasionally break the physical connection between genes on the same chromosome. This mechanism is crossing over, and it occurs between homologous chromosomes. Okay, so if we want to determine our recombination frequency, what we do is we take the total number of recombinants, which is, I think that says 291. This is really small on my screen for some reason. And they divide that by the total number of offspring, and they get 17%. Okay, so that's a really small recombination frequency. So what that basically leads us to conclude is that these genes were close together on the same chromosome. So we say that they are linked. Recombinant chromosomes bring alleles together in new combinations in gametes. Random fertilization increases even further the number of variant combinations that can be produced. This abundance of genetic variation is the raw material upon which natural selection works. Alfred Sturtevant, one of Morgan's students, constructed a genetic map, an ordered list of the genetic loci on a particular chromosome. Sturtevant predicted that the farther apart two genes are, the higher the probability that a crossover will occur between them, and therefore the higher the recombination frequency. A linkage map is a genetic map of a chromosome based on recombination frequencies. Distances between genes can be expressed as map units. One map unit represents a 1% recombination frequency. Okay, so this is one such linkage map, so you can see that the recombination frequency between these two genes is 9%, recombination frequency between these two is 9.5%, and the recombination frequency is 17% between these two, because these two are further apart than these two are, or these two. So the further apart they are on the same chromosome, the higher the recombination frequency. Genes that are far apart on the same chromosome can have a recombination frequency near 50%. Such genes are physically linked but genetically unlinked and behave as if found on different chromosomes. So when we're doing problems to determine whether genes are linked or unlinked, 50% is the magic number. Less than 50% is going to be linked genes. Greater than 50%, 50% or greater, is going to be unlinked. Sturtevant used recombination frequencies to make linkage maps of fruit fly genes. Using methods like chromosomal banding, geneticists can develop cytogenic maps of chromosomes. Cytogenic maps indicate the positions of genes with respect to chromosomal features. So this is a partial genetic map of the Drosophila chromosome. Large-scale chromosomal alterations in humans and other mammals often lead to spontaneous abortions or miscarriages or cause a variety of developmental disorders. Plants tolerate such genetic changes better than animals do. In non-disjunction, pairs of homologous chromosomes don't separate correctly during meiosis. As a result, one gamete receives two of the same type of chromosome and another gamete receives no copy. Non-disjunction can occur in two places. It can occur during meiosis 1, like here, or meiosis 2, here. So if it occurs during meiosis 1, then notice what happens here. Okay, we already have a cell with too many chromosomes and a cell with not enough chromosomes. As a result, after meiosis 2, we end up with 3 in one of the gametes, 3 in another gamete, and then 1 and 1. So we should have 2 in each one of these. So if non-disjunction occurs during meiosis 1, then all of the gametes will have an incorrect number of chromosomes. If it happens during meiosis 2, then notice that 2 out of 4 of the gametes will be normal. Okay, so only 2 of these will have the incorrect number of chromosomes. Aneuploidy results from the fertilization of gametes in which non-disjunction occurred. Offspring with this condition have an abnormal number of a particular chromosome. A monosomic zygote has only one copy of a particular chromosome. A trisomic zygote has 3 copies of a particular chromosome. Polyploidy is a condition in which an organism has more than 2 sets, complete sets of chromosomes. Triploidy is 3 sets, tetraploidy is 4 sets, polyploidy is common in plants, not in animals. Breakage of a chromosome can lead to 4 types of changes in chromosome structure: deletion, which removes a chromosomal segment; duplication, which repeats a segment; inversion, which reverses the orientation of a segment within a chromosome; and translocation, which moves a segment from one chromosome to another. Okay, deletion, we should have a D, we don't have a D. Inversion should be A, B, C, D, A, D, C, B. It's backwards. Duplication, we should only have 1 copy of B and C. Here we have 2 copies. Translocation, a piece moves from one non-homologous chromosome and switches with another. So notice the difference here. This should be A, B, but instead we have MNO. This should be MNO. Instead we have AB. A diploid embryo that is homozygous for a large deletion is likely missing a number of essential genes. Such a condition is generally lethal. Duplications and translocations also tend to be harmful. In inversions, the balance of genes is normal, but phenotype may be influenced if the expression of genes is altered. Alterations of chromosome number and structure are associated with some serious disorders. Some types of aneuploidy upset the genetic balance less than others, resulting in individuals surviving to birth and beyond. These surviving individuals have a set of symptoms or syndrome characteristic of the type of aneuploidy. One example of this is Down syndrome, trisomy 21, where the individual has 3 copies of the 21st chromosome. Frequency of Down syndrome increases with the age of the mother. Okay, so here's a karyotype, which looks at an individual with Down syndrome. You can see that there are 3 number 21 chromosomes. Non-disjunction of sex chromosomes produces a variety of aneuploid conditions. Klinefelter syndrome is the result of an extra chromosome in a male producing XXY individuals. Females with trisomy XXX have no unusual physical features except being slightly taller than average. Monosomy X or XO is called Turner syndrome. Females with Turner syndrome are sterile. It's the only known viable monosomy in humans. Syndrome Cri-du-chat, cry of the cat, results from a specific deletion in chromosome 5. Child born with this syndrome is mentally retarded and has a cat-like cry. Individuals usually die in infancy or early childhood. Certain cancers, including chronic myelogenous leukemia, are caused by translocations of chromosomes. So here we have normal chromosome 9 and a normal chromosome 22. This is showing a translocation that's associated with leukemia.